The reason why what we do is important is because we have a problem in the field of human genetics. And that is that although we can now quickly, cheaply, and easily sequence an individual's genome, we don't know whether or not the variants in the genome that we find in the genome um, are likely to cause disease or are completely harmless. And there's a big need to be able to tell the difference. Because if you find a harmful variant, then that could lead to shortening the time to diagnosis and shortening the time to treatment. And for urea cycle disorders, as your community knows, the shorter the time to treatment, the better patient outcome will be. And so our work is critically important for determining whether or not how impaired a variant that one might find in a human genome is or isn't. At some point in the very near future, every newborn is going to be whole genome sequenced at birth rapidly. So the other thing technologically that's happening is that there are new technologies for sequencing that are fast and cheap. And so there have been studies where people have gone from time of taking a blood sample to making a genetic diagnosis in 8 hours. Somebody here in Seattle has done it in 3 hours. So rapid newborn sequencing, not just of very sick babies in the NICU, but also of just general newborn screening, that is going to happen. And those technologies are happening. The important studies are happening. The important conversations about social, legal, and ethical are happening. And so that is going to happen. So now the problem is, what you're going to find is a lot of variants of uncertain significance. This is like the biggest thing to happen in genomics in like the last 10 years. And I was so excited about it. And I was like, we have to do this. And so I was looking for an excuse to do it basically. And so so the fact that we get to do the technological things that we're excited about, that we're good at, and also that we get to have an impact, like as a yeast geneticist, I could work on, and we have worked on, understanding basic science. Like we use yeast to understand how cells function and basic processes and how to do genetics better and how to develop new techniques and all of that stuff. And that's good. And we get excited about that. And I love doing it. But this whole thing has let us do it in such a way that, we have physicians and sometimes even families thank us for what we do. Um, and that's just incredible. So this is the most rewarding work that I have ever done and that my lab has ever done. It sounds, kind of incredible, but it was really just chance. So we had been studying an ultra rare disease, and we had kind of taken our analysis of that to kind of its logical conclusion. And so I realized that our lab could inform lots of other human diseases. And I was looking through lists of metabolic pathways that were similar between yeast and humans, and we have ways to kind of analyze that at the sequence level. Um, and I went through one pathway, and I was like, all right, this looks great. This looks like we'll be able to do it. It's a nice big pathway, and this looks great. And I was like, all right, what human diseases are associated with this pathway? And there weren't any. So
Okay.
so I moved on to the next pathway, and that pathway was the synthesis of arginine, um, which your readers will know that is what underlies the urea cycle. So we were, starting to do this, and we were just working kind of in a vacuum and doing our technology. And then my colleague, Claudia Carvalho and Andrea Grotman, were sitting on a panel together, and they started talking. And Claudia made the connection of, Andrea, you study urea cycle. Um, you should talk to my colleague who's got this new technology. And so kind of the rest is history.